And here we are with our first keynote. I have the greatest honor to present Arcadi because he remembers 12 years ago I was presenting him. I don't know what was the month, but 12 years ago I was in university and we organized some sessions about evolutionary biology, and was during that symposium when I hear Arcadi, when I decided I wanted to do bioinformatics. And yes, and that's why I'm here today. So thank you very much, Arcadi, and I hope you're going to hear a great presentation about him. I'm going to present his work, which talks by itself. Dr. Arcadi is doctor in genetics and has centered his scientific career towards evolutionary biology, being appointed research professor in 2006, professor in genetics in 2010, and director of the Department of Experimental and Health Science in 2013 in the Institute of Evolutionary Biology in the University Pompeu Fabra. In 2016, his career took a twist to become the Secretary of Universities and Research of La Generalitat, returning to UPF in 2019 and becoming the director of the Pascal Maragall Foundation in 2020. Moreover, he's an active mentor, teaching at undergraduate and graduate level, and having supervised 10 PhDs theses. Lastly, Professor Navarro is also committed to communication science to the public, and proof of this are his awards of the 11th European Scientific Divulgation Award and the Casa de las Ciencias Scientific Divulgation Award. I'm sure you are going to have a great presentation here, and I leave you with the audience, Arcadi. Thank you very much for being here today.
Many thanks, Sarah. It was a real pleasure seeing your face when we went backstage and immediately recognizing you. What I was not aware of is that I was a tiny bit responsible of your current career status. My feelings are mixed. I'm really very happy because I think bioinformatics is very cool, but also, who knows? I mean, if things go bad, please don't go around saying it's my fault or stuff like
this.
No,
of course not.
Okay.
Yes, thank you for introducing me to this great work.
Brilliant. So let me share a screen.
Yeah.
Let's see if I can find the proper screen I want to share. And this should be the screen I'm sharing. Is that the case? Can you see the screen?
Yes, perfectly.
Perfect. Okay. So, well, thanks to the organizer, sorry. Actually, when I accepted coming, I didn't know it was Sarah. So I hesitated a bit, right? But only a tiny bit. I think it's excellent that there's regional student groups and that the student groups organize meetings like this. I do remember when I was growing up that there was not such an active and thriving community as there is now. So well, thanks for being here. And please, I'm told that there will be some time for questions afterwards. But if there isn't, feel free to drop me an email, right? Don't hesitate if you want to ask. I'm easy to find over the internet. You just Google me. I'll be there. I cannot promise an immediate reply because there's many mails every day, but I'd be really very happy to be in touch with you afterwards, right? So as to today, let me start by saying that I work here. I work in several places, but this is my favorite, right? Because obviously this is in Barcelona. You get to see the sea every day, right? And of course, I never work alone. So there's a bunch of excellent people. The photos are a bit outdated because since the pandemics, we could not arrange for proper photo taking. But some of the stuff I will be presenting today is the result of the work of some people, actually many people who are not here, right? And today I will, as I was saying in my title, I will tell you about how can we bioinformaticians try to do more and better science, not only for ourselves, but for the world. So I will first tell you about what big data is or maybe. I will tell you about the sharing dilemma. I'll tell you what can we do, actually, what we are doing to actually contribute to the sharing dilemma, I will give you a very quick example. I will just tiptoe over it, an example of what you can do if you share data. And then, of course, I'll have some key conclusions, right? So let me start by this slide. What do all these images have in common? Well, what do they have in common is that they are all information, they are all data that we can access mostly through the internet, right? You've got maps, you've got your personal data, your shopping lists, your health data, you've got your wearables, you've got, of course, hundreds, thousands, millions of videos of kitten in the internet. So all this is big data. And big data is very big, right? It's very difficult to get these estimates, but in the world, since the origin of humanity, lots of information has been generated from the beginning of written history. Actually, not even written history. Before that, recorded time. This counts the Altamira paintings, right? Until 2003, humans had generated, it's estimated, about 5 billion gigabytes of data. In 2011, already 10 years ago, the same amount of information was created every 2 days. By 2013, every 10 minutes. And the best estimation for last year, there are no estimates for this year, as far as I know, is that about 1.7 megabytes of data are created every second by every person on Earth. This is 13 petabytes per second, or 1 exabyte per minute, right? This is a crazy amount of information. And admittedly, most of it are videos of kittens on YouTube, or perhaps photos that we take with our telephones, and that we never see again, right? There's these fantastic statistics by Apple saying that most of the photos that are uploaded into iCloud are never opened again, not even by the owner, by the person who took them, right? So there's these big computer farms out there in the wild hosting data that will never be accessed again. But that's another issue. This is big data, right? And this is growing so much that we have a very clear Pareto. 90% of the data in the world today has been created in less than the last year, which is amazing. And again, this data comes from everywhere. As I say, sensors used to gather climate information, your posts to social media, digital picture videos, GPS signals, whatever, right? This is big data. And well, if we were in an informatics conference, I will focus to any kind of big data. But I would focus on one part of big data because we are in a bioinformatics conference, right? And I will mention that there's a very small fraction of this data that belongs to the field of medical genomics. And of course, this is a tiny fraction if you consider the huge amount of data, but this is still an enormous amount of data in itself, right? This big data, this fraction of big data, it's also big data in a sense. And it's a data that is relevant for human health, for the biomedical sciences, right? And that's the kind of data we usually deal with, right? And this is a deluge now, but it was not always this way, right? Back in 2001, 20 years ago, we had the presentation of the Human Genome Project, the results. And this was an amazing conference because not only the first draft of the Human Genome was presented by this, one of my heroes, right? Even though I don't like his views on religion, but it doesn't really matter. Francis Collins here, who only a couple of weeks ago stepped down as the NIH director. And here you had Clinton and Blair presenting this, right? And it was a single press conference held at the two sides of the Atlantic. And this was the first time that some video system was installed. Now we can do this again with our telephones. We all can, Zoom or FaceTime or whatever. But now, at the time, sorry, this was almost as innovative as the human genome itself, right? And when this started, only, a tiny amount of data was available, a single genome when you think about it. But since then, there has been a deluge of information, a huge amount of information, like, a flood of information. We have had many genomes from literally thousands of individuals, from literally thousands of species, epigenomes, whatever, you name it. There's amazing progress that has taken place over the last 20 years. And this data, a fraction of this data, is relevant for human health. Of course, you've got other species, right? I will not cover this today. But as I say, a fraction of this data, together with relevant medical data, together with relevant socioeconomical data, is actually the main source of research, the main focus of research, sorry, on the biomedical sciences nowadays. That's the kind of data people are generating, and that's the kind of data people are analyzing, right? And this data has a problem. And the problem is our sharing dilemma. What do I mean by a sharing dilemma? Well, you may not be aware of this, right? But there's the issue of human rights. If you look at the Universal Declaration of Human Rights, there are perhaps conflicting rights. So there's one right, I think it's Article 19, but I forget now, that states that all humans have the right of participating into scientific progress, right? To benefit from the fruits of science, particularly if the science has been paid by the taxpayer, by us, because it has been funded and it has taken place in public organizations, right? But then there's another human right, which is the right of keeping your private information confidential. And of course, it is easy when you think about it. It is kind of simple to keep your X-rays confidential, right? You can share your very X-rays around because there's no way to trace these X-rays to you once your names have been erased. If you go walking in the street and somebody does another X-ray, it will not be possible unless it's your teeth, for instance, right? It will not be possible from an X-ray of your lungs to identify you. Actually, well, if you have a tattoo, perhaps because tattoos appear on X-rays, but there's this rule: before sharing an X-ray, you erase the tattoo, right? This is not the case with genomic information. This is not the case because you cannot erase personal information about somebody's genome without actually erasing all the relevant information that there is in that genome. So the community is split, right? You have to keep this information private, but also you have to share it, because by sharing it, you can generate more science. You can generate meta-analysis. You can reanalyze data using new ideas, using new algorithms. So sharing is important, right? How do we solve this dilemma? Well, when this dilemma started being obvious back in 2007, 2008, several different institutions decided that data should be made fair, should be made findable, accessible, interoperable, and reusable. Actually, this is the invention of a colleague, right? Of a guy from the Netherlands who actually came up with this beautiful acrostic. In order for data, in order for humanity to benefit from scientific data, this data needs to be findable. You need to be able to detect it. Perhaps it's a store in Japan. It doesn't really matter. You have to find it out. You have to be able to access this data, right? If it's in a hard drive, in a shelf, it's really not relevant. This data needs to be in formats that are interoperable. It cannot be done in a formal, it cannot be stored in a format that no one else can use, right? And of course, the data in itself, the metadata and the way data has been gathered needs to be such that data are reusable for other analysis, right? So this is my own position, the dilemma. I think that scientific data must be fair, and I myself lean towards the human right of benefiting from science than to the human right of privacy. But that's another issue. And of course, it's not my position that matters. It's the position of our society. And our society, on average, wants privacy, right? So what to do with this? What can we do to solve this dilemma? Well, back in 2008, already, different governments started proposing several solutions, right? And in particular, for the case of genomic data, you've got the solution that was created by the American government, the database of genomes and phenomes. Here, it's essentially a database in which researchers that are funded by the NIH put their data at when the paper is being sent for publication, their GWAS data, the data from association studies, cases and controls, people with type 2 diabetes and people without type 2 diabetes, their genomes get compared, you publish a paper, you discover new genes and new variants, and before publication, you put your data in this database. So other people can access this data, right? And the European Union at the EVI created the European Genome Phenome Archive, right? Which is more or less the same thing. And the concept is very simple, really. And you've got also the Japanese database, which is really very small. The the European Genome Phylum Archive is an archive for human genomic data that should have control access because it's private data, it's personal identifying information, data generated by research centers all over the world, and then there's a data access committee that controls access to the data. And the EGA controls or coordinates all these more than 1,400 now data access committees, because each data set has its own data access committee, right? And the thing here is that because the original investigators had to abide to certain rules to get data from people, they had to abide to the general law of the land, and they had to abide to the informed consents, the donors of the data generated, right? And this implies accepting some legal paperwork yourself and your institution. This implies, for instance, monitorization by an ethical committee and so on and so forth, because the original producers of the data accepted all that. Everyone that will download the data actually has to accept the same rules, the very same rules, right? When the EGA was created, and this is what it does, it's essentially me telling you that it's a very secure system, as secure as a Swiss bank, so the data are properly encrypted, stored in a vault without access to the internet, and so on and so forth. It was a project that back in 2008 was a project by the European Bioinformatics Institute, right? But since 2013, it became a joint project between the European Bioinformatics Institute and the Center for Genomic Regulation in Barcelona, in this building that I have shown you before. But of course, the data are not here. The data are both that there's a copy in Cambridge, in Kingston, near Cambridge, and there's a copy in Barcelona. The copy in Barcelona is sitting in the Barcelona Supercomputing Center, right? So it is a joint project. And it's a project that has grown enormously, right? It started in 2012 with a few files and not a lot of terabytes, but as you can see, it has kept growing, and by now it has almost 15 petabytes of data, right? With thousands and thousands of study. This big jump here is the UK Biobank, for instance, right? And essentially, all these millions of files, all these thousands and thousands of megabytes and terabytes and whatever, essentially are different studies, and some of them are really very famous, right? Every major study that you have heard of is actually hosted somewhere else, somewhere in the EGA. So the EGA actually is able to steward it and distribute it to bona fide researchers from good institutions all over the world, right? And who is downloading this data? Well, out of this data, we daily distribute about 50 terabytes of data to anywhere in the world: Switzerland, Japan, Canada, China, Spain, and of course the United States and the United Kingdom, where most of genomics research takes place, right? But many other countries, all this green thing here is literally tens and tens of different countries, right? And whose data are we taking care of and distributing? Well, pretty much everyone's data, right? All our data sets come from different countries in the world. And interestingly, there's lots of them that come from the United States. And that's a funny thing, right, about sharing, because sharing is important. There is a mission. But sharing is also publicizing your data. If you make your data findable, other people may be interested in the research you do. They may ask for your data and they may get your data, but they also, if they know it is your data, they may also ask you for collaboration, right? And interestingly, in the United States, what happens is that in the database of genomes and phenoms, the researchers that originated the data lose control of it. They cannot track the data. It is the government that decides everything. There's a single data access committee. In the European Union, however, you don't. As a researcher, you can always know who is using your data. And that's lovely publicity and lovely opportunities for scientific collaboration, right? And that's why the American researchers tend to save the data from us. American researchers for 144 different institutions, they tend to send the data to us unless it is data that they have generated funded by the National Institutes of Health, by the NIH, because in that case, they have the obligation of putting it on the DB guide, right? Now, this is all very well, but to me, the most important thing is, what is this good for? What do we achieve by doing this? Well, imagine yourself in the scenario that you have to put together 20 different papers, 20 different studies, data from 20 different studies, that all of them studied, I don't know, colon cancer, because you want to run a meta-analysis with all this data using a new algorithm that you have developed as a PhD student, right? What should we do if there was no EGA? If there was no EGA, you would have to contact 20 different research groups, and you would find a variety of situations, both logistically and legally. In logistic terms, you would find that some institutions, large institutions, probably had the data handy, and they were, eager to give it to you. Other institutions, logistically, perhaps the PI doesn't even know where the data is. Perhaps the postdoc who managed the project left already, and perhaps the data are sitting in a hard drive somewhere, right? So logistically, you may have lots of problems to get access to these 20 different studies. But also legally, you would find 20 different jurisdictions, perhaps 20 different countries, and you would have to deal with things in 20 different languages, because you would have to deal with lawyers, right? And sometimes some people would make it easy for you, but other times some people would not even want to waste their time dealing with all the petitions of data access, right? So many papers, many scientific projects, many discoveries would be impossible. Just perhaps in some cases just delayed, but in many cases just utterly impossible. Thanks to the EGA, most of the things you don't have to do. You submit a single application, and it is the EGA that deals with different studies and with the legal permissions and so on and so forth. For instance, we have generated an ontology of all the conditions that are implemented in informed consents. So the informed consents are machine readable, and when we read your project, the things you want to do with the data, we can know whether every single informed consent allows for it, right? There's data sets that have very restrictive informed consent. Some of them are for research, but some of them are only for instance, pediatric research, right? So all this has to be taken into account. And we are so successful that every year, this is non-cumulative data, thousands of papers are published that actually, in some way or another, have made use of data coming from the EGA, right? In some cases, it's just the result of the paper, and data has not been downloaded, but in thousands of cases, in 2021, this is June data, so it's half a year. We will probably go beyond 30,000 papers only this year that have been made possible up to a point thanks to the EGA sharing data, right? Now, is this enough? I would say it's not, right? Because of course, think about this, right? Data has to be applied for, you have to write a project, you have to have an informed consent read, you have to have a ethics committee monitoring you, and so on and so forth. And there's different laws. So all of this fortunately happens in a global scenario in which the EGA is part of a very rich ecosystem in which institutions such as the Global Alliance for Genomics and Health or Elixir in Europe actually do lots of interesting stuff to make data share possible, right? For instance, one thing that actually we ourselves invented is something that we call the beacon, right? So before spending 2 months doing paperwork, before accessing a data set, you'd better know whether this data set is interesting for you or not, right? Imagine that you are interested in knowing whether there are individuals in this particular study of, I don't know, 2,000 people, whether there are individuals that have a certain mutation in a certain position in a certain chromosome. If the answer is yes, you want to see what those individuals are that have this particular mutation. But if the answer is no, you don't have to waste your time applying to your data set, downloading it, and so on and so forth. So what we are doing is making a good, enormous effort to distinguish discovery from sharing. And what we have now is a series of beacons that are sitting on top of every data set in the world that has joined the initiative. And they are such that you can ask, hey, do you have, I don't know, a patient that has a deletion in chromosome 9 in a juvenile female with glioblastoma? And perhaps the answer is yes. And then you do the stuff, And perhaps the answer is no, and then you look at something else, right? So we are trying hard. The different data sets in the world, sometimes they are in DGA, sometimes they are not, um, like beacons, so they are easier to discover. They are fairer, right? And of course, we have created a beacon network, so you don't have to you don't have to talk to every beacon separately. You can do a single query, and the network will distribute you all over the world, right? And this, in the European context, is very important because some of you may have heard about the Beyond 1 Millions project. And actually, the Beyond 1 Millions project has the interesting feature that it relies a lot on solutions, on software that has been produced by the EGA. It relies a lot on the federated DGA, it relies a lot on Beacon, and so on and so forth, right? I can give you more details later, right? Because actually we are moving to a model in which the EGA is all centralized with only a couple of computers holding all the data, to a model in which different nodes can be open. I don't know, a German node, a Finnish node, and it's such that the data that the jurisdiction wants to remain in this particular jurisdiction can still be discovered because different countries, different Elixir nodes are going to be able to join efforts doing that, right? And this is, of course, something that we have been doing not only ourselves, not only in Barcelona and Kingston, but also with the collaboration of many different people in different countries, right? So this is an important effort of data sharing that has the declared goal of having a number of genomes available by the end of 2022, but that involves a huge coordinated effort in data sharing, right? And of course, this is for the Spanish people in the audience. This is a funny thing because in many of these countries, there has been considerable funding for personalized medicine initiatives. But Spain, until very recently, didn't have a personalized medicine initiative. Still, Spain actually funded a couple of very relevant softwares like Beacon, RDConnect, the EGA itself, that have become central in this endeavor, right? Anyway, I would also say that this generates interesting possibilities for young bioinformaticians, because of course, if the data are in a federation rather than in a centralized site, if the data out of the legislation in some country needs to have a copy in this country, in the original jurisdiction, so, there are two things that happen. First, it would be a waste of resources to have several different copies in different places kept securely. You keep the copy and the backup in the original jurisdiction. That's why you have a federation. But second, you start having a pressing need to carry out federated analysis. That is, rather than bringing all the data to a single place where there is the algorithm, you send special algorithms to different places where the data are sitting and then reanalyze everything. And that's an interesting concept, right? But I will not dwell into this today. And of course, all these data sharing initiatives have had a very nice, I would say, a stupendous use during the COVID-19 pandemic. If there were if they had not been sharing initiatives by early 2020, the response to the pandemic would not have been so quick. Actually, we keep saying that we had the vaccine in only 1 year and a half. To be precise, we had the vaccine in more or less 48 hours. Only that, of course, it took 1.5 years to do all the testing, right? But because of data sharing structures, the first sequences of the virus that were obtained in China were sent to the relevant companies, and the first vaccines, the first targets, were designed in less than 2 days, and they were already being produced to run tests, right, to run clinical trials. So data sharing is indeed a very powerful thing to do better science, to do more science, and to do all of this better and more science quicker, right? Let me give you a quick example of all this. And this is a bit of research I have been involved, in which I am involved myself, right? And what I would say is that senescence implies deterioration of function, and it's a problem in general terms, right? If you have to select a headline for the last century, what would you select? That humanity defeated the Nazis, that we managed not to kill ourselves in a nuclear war? Actually, The most remarkable thing that has happened over the last century is that average lifespan has moved from about 30 years to about 70 years. And this is not because of children not dying. Children not dying is an important fact. Infant mortality has been reduced, but the most important effect is that people have stopped dying when they become older. So we have moved from a population in the early 20th century of about 2 billion people to a population of almost 8 billion people in the planet, just because people don't die, right? And people who don't die become senescent. They become old. They become fragile. And this is an important issue, right? And let's face it, humans tend to be senescent. Nowadays, senescence is the leading cause of death because you can die out of many different reasons, many different diseases whose risk increases with age, right? So senescence is a crucial public health issue. We need to know what we can expect in the near future, and we need to know how we pay for it, right? We need to know whether people will become more long-lived and will have a longer health span, not only a longer lifespan, but a longer health span, just as it has happened so far. Today, I'm 50 something, right? This is being like 40 something 30 years ago, because essentially I'm better fed and have better doctors. I have more healthy lifestyle, right? But not only it's an issue of public health, but also it's a very exciting scientific matter. How do we decay with age? And also, why are we senescent at all, right? And this is a pressing issue because, let's face it, humans are senescent. Perhaps there are some exceptions, right? And this guy is retiring soon, and he still looks the same. And for the non-Spaniards, these photos are almost real, right? And of course, also for the non-Spaniards, there's some cases that are not so funny. That are some sinister cases of people who actually don't seem to age, right? But let's face it, humans age, and I have a clear proof here, right? This is me when I met Sandra a few years ago. But anyway, the reasons for senescence are unknown or are not fully understood. The most immediate reasons, I don't know, free radicals or inflammation or so on and so forth. Of course, the molecular research is really well advanced. But why are species senescent at all, right? So there are two theories for that are more than half a century old. The mutation accumulation theory, it comes from Peter Medawar, who was a Nobel Prize, and essentially it states that if you have a mutation that has a deleterious effect on the organism, when the organism is young, well, this mutation that destroys the organism before it can reproduce, that harms the organism because before it can reproduce, will be kept under control by natural selection, right? Because if you cannot reproduce, you don't make it through the next generation. But if there's mutations that harm the organism once the organism has reproduced, if there are mutations that actually do bad things to old people, these mutations cannot be removed from by natural selection from the population, right? And actually, there's this other guy, George Williams, who actually proposes another thing. He proposes that let's imagine that there are mutations that are bad for the old, but good for young people. I don't know, a mutation that when you're young allows you to accumulate lots of calcium in your diet, right? This is good because then your bones are more resistant. But then when you become old, once you have already reproduced, this mutation may increase the risk of arteriosclerosis, right? Which is a problem. But hey, you have already reproduced. The mutation is successfully already in the next generation. So these mutations may accumulate and explain senescence, right? That's a long story, but this was proposed 50 years ago. And the idea is that we have all this richness of data, association studies that are published by thousands of groups all over the world. And we can translate these theories in terms of health and also in terms of the mutations and the genes that are being identified, right? In terms of health, do we get sick when old because natural selection is less efficient in eliminating things that damage you when you're old? Or do we get sick when old because natural selection favored health when young at the cost of screwing you up when you were old? And if you think about mutations, are there risk alleles for late onset disease that are at higher frequencies in the population and have larger effects than risk alleles for early onset disease, of course? And of course, are there out there mutations that have this pleiotropic effect that George Williams proposed, that are such that are mutations that protect you from early onset disease? And actually, at the same time, the same mutation, the same allele, increases the risk of some late onset disease, right? If this is the case, if these mutations exist, we may have been able to prove that these two theories that had been studied in many organisms also apply to humans, right? And in order to do that, of course, one had to get all this data. And that's the main point here. The main point is that only this kind of sharing policy, this kind of sharing infrastructure allows this kind of research, right? We got this data, and I'm not going to go into details, but of course, I would be happy to have correspondence about this. We find that when you look at allele frequencies, risk allele frequencies of diseases at different ages, this is the age of onset of the disease, you can see that late onset diseases have different edges of onset, right? This is the same diseases all the time. Every point is the same group of diseases, only that we classify because, of course, we need to have the data speak by itself. What's old, what's young, right? At my place, opinions are divided. My mom thinks I'm a very young guy, and my kids thinks I'm an old geezer. So what we did was to get all the diseases, all the associations that we knew, and do the test in every threshold. And what we can see is that when you consider that beyond 10 is old and earlier than 10 is young, which is of course true, you very clearly see that there's a significant difference. And same thing until 30 something. Then, of course, if you start saying that beyond 50 is young, you lose significance. But the patterns are still there. And the same thing for pleiotropic mutations, right? Don't care too much about what this graph means. It means that if you think that the change between old and young age, young and old, is between 40 and 50, then you find an excess of mutations that you can see here that are such that protect you from early onset disease at the cost of increasing the risk of late onset disease, right? And that's beautiful because this is us interrogating databases of disease. But there's plenty of other data sets that fit with our observation. I mean, menopause started at 40, 50, and then our ancestors rarely lived beyond age 40, which essentially means that what we are explaining makes sense. We are getting all these databases from all over the world. We are merging them for analysis, and it fits with other observations that are independent. And there's a bunch of papers in which the genes that we detected that harbor these pleiotropies have been shown to be genes that are related to aging, to senescence, right? And that's a project I would pose that it's a beautiful example of what one can achieve by sharing data. By using data shared by others, we have been able to first find new uses for the data. Those uses, studying senescence, are something that people did not have in mind when they generated the data, I don't know, on diabetes or on colon cancer or on whatever, right? They didn't. We have been able to tackle fundamental biological and evolutionary problems related with pressing health issues. And we have been able to make progress, particularly in the area of senescence, highlighting the importance of pleiotropy, of relationships between different genetic relationships between different diseases. All this would have been impossible unless people were sharing data, right? So what's my conclusion of today's talk? It's very simple, really. I have been repeating this message over and over again. If you want to have better science, if you want to have more science, and if you want this to happen quick, because knowledge saves lives and we want knowledge generated quickly, there's one thing we should do. And the thing we should do is sharing. And that's about it. Thanks very much.
Thank you very much, Arcadi.
My pleasure.
Great. Okay, so we're going with the questions. I'm going in order. First, one from me. I'm very interested in EGA and in ENA because I mostly work with microbiology data. So more ENA than EGA, but would an institution be able to deploy a local EGA if they have the resources, or it's going to be selected institutions for this?
There's going to be a selection because in order to deploy a local EGA, you can do two things, right? You can get our software. It's open source, so you can do whatever you want, but then you will not be able to share data with us or to use our image and brand unless there is an agreement, right? An agreement between CRG and EBI and the institution in question. And we have several of these agreements being studied now by different lawyers. Now, I didn't get into details about the federated EGA, but there are like two different things, right? You can have a full federated node, and this implies a serious commitment of resources. You need to have a help desk, you need to have people who are developers, you need to commit to stay at least 4 years in service and so on and so forth. So people are rarely going to be able to do this unless they have a mandate from their institutions or from their governments. But there's another more lightweight thing called EGA community, EGA community. And this EGA community, it's far more simple. You only make sure that your data have metadata that are such that can be discovered within the EGA databases. And that's far more simple. Both are possible. The first one requires resources. The second one, you can light for a few months and then you can decide to stop. So it requires some resources, but not so many. So bottom line, we are trying to make this in a way that the more institutions, the more countries that participate in this, the better. So we try to make things easy for people.
Okay.
Not that we succeed a lot, but anyway.
Yeah, it's really difficult to do that. We are going to have a lot of data from surveillance from different microbes, including COVID-19 and so on, because we're the center, the National Health Center, and that makes surveillance for the government. And we are trying to see how we can store all of the data in an organized way. And I think if there are exports that like ENA and EGA, we can use that, of course.
Of course. So one important thing is that if your data is such that needs to be kept private, EGA is a good solution for you because it has all these layers of security. But if your data is such that you only need to keep it under control, you need to know where stuff is, but there's no privacy issues, then perhaps you should go to the ENA, to the European Nucleotide Archive, because it's a far more simple setup. You don't have to keep things secure because, no problem if there's a leak. Your genome is not going to be seen by other people. Anyway, that's another matter. I just was checking now. I don't know if I have your email, but let's email after this. Anna,
okay, let's discuss, yes. Next question. That's an outstanding talk. Thank you so much. Beacons are very useful, but do they query over already processed data? What if data could be realized to improve the results from a beacon? For example, NSG data realignment to improve variant coding.
So at the moment, what beacons do is very simple. They started even simpler. They started being applicable only to databases in which you could ask very atomic questions, very simple questions. As I said, do you have this particular variant in some individual in this particular chromosome? And nothing more complex could be done. Now we are moving towards something that is more, we call it the clinical beacon, that it allows to also query things that are more sophisticated, several variants at the same time. It allows to query the profiles, the phenotypic profiles of patients and so on and so forth. There's a limit though. You should make sure that the queries you make don't reveal private information. So if, I end up, is Arcadi Navarro's genome here, there? And the answer is yes, this is a bad thing, right? So the equilibrium is kind of difficult. So we try to make sure that we strike a good balance. And that's a moving target because, of course, more sophistication keeps appearing. As to realizing data to improve the beacon is kind of complex because the beacon in itself only will tell you, it's designed to tell you this bunch of data has something interesting for you. The handover is usually the reference number of the study so you can apply to it. The handover is perhaps the email of the PI that controls the study, so that owns the study, so you actually deal with that PI and so on and so forth. What we're trying to do is not to ease things at this level. At the level of Beacon, we try to ease discovery only, but at the level of databases, what we are trying to do, and we have some pilots already that are fairly good, is that rather than downloading the whole database, you may be able to download it, and first this needs process, a given chromosome or a given gene that you are interested in. And actually, ideally, you should be able to do this on the fly. And this implies some reanalysis. It could be lovely that rather than downloading the raw data only, you could, for instance, download some data, realign with using some algorithm, or impute it using some reference panel. That would be great, right? And we are trying to slowly add these improvements. So yeah, there's lots of things that we can do, but so far we are doing it more in the area of sharing, and in the area of Beacon, what we're trying to do is to ease the discovery, not the reanalysis. Hope this helps, Javier.
I'm sure. Okay, following, Ines asked this. Great and very useful knowledge on data sharing. Thank you, Arcadi. Considering data sharing privacy from a world's perspective, how can global agreements be reached, given that different cultures weight privacy versus common benefit differently?
Well, all the questions were good, but so far they were all scientific. This is a good and darn difficult question, because this is not something that I can answer as a scientist. I can try to answer this as a policymaker. I was a policymaker for a while, and I think that what we need to do is to do essentially two things. I'm going to be very idealistic, but I have to be short. First, different governments, different institutions have to understand that science is not the project of a research group or an institution or a country. Science is a global project of all humanity. What's the difference between the science made in China and the science made in Argentina? There's no difference. It's all science. And in order to be science, you have to share. There's no science without sharing. There's no Einstein without Newton. There's no Newton without Kepler. There's no Kepler without Tycho Brahe. Because all these people share their data, and new people that were better positioned could reinterpret the same data from a different angle, right? So sharing is fundamental for science, and governments and institutions need to understand this. The second thing, besides preaching, you have to incentivize. And I think that what we are doing is trying to solve a confusion. 10 years ago, when you went to a doctor and told the doctor, hey, you need to share your data, the doctor immediately said, I'm not going to share data, because this data is what I need to progress, to have my career progress. I'm not going to share this data, because then other people will do the papers and I will not be promoted. Now what we tell this person is, hey, you have to make your data discoverable. So you have a big loudspeaker to tell the world that you have this data, and this makes you more interesting. You have more contacts, and then you have a better career. So besides naming and shaming, besides preaching the institutions to make these things compulsory, you also, which is working top down, you also have to work bottom up and convince people, individual researchers, that if they share data, it's going to be beneficial for humanity, but also beneficial for them, right? And that's the type of thing that we are trying to do right now. I would say that sometimes we fail spectacularly. Like China changed the law like 2 or 3 years ago. 2 years ago, actually, they made a very tough privacy law, and it's now darn difficult to share Chinese data out of China. And I'm not going to be the one talking to Xi Jinping and convincing him that this is wrong, right? We have to do otherwise. But we need to do this, because otherwise we are stopping progress.
Yeah, it's really difficult. And explain doctors or scientists that they have to share data, it is still a challenge most of the times. Depending. Okay,
I see cards and things.
Yeah,
Thanks very
we
much.
have
This
more
is
questions, but we are on time. So if anyone wants to ask Arcadi, please write him. I'm sure he will answer without any problem. And thank you very much, Arcadi. It has been
my
great,
pleasure. It
as
it
always.
has been great. You're too generous, Sarah.
No, I'm not.
Enjoy yourself. Bye
Bye.
bye. Bye everyone.
Okay,
Cheers.
I'm going to end the session and start the new ones. See everyone in a couple of minutes.